All right, great. Thank you everyone for coming. My name is Aaron Wenger. I'm working in product marketing at PacBio. I'm just going to give you an update today on some of the new products that we've announced this week. I'll start with a basic overview of our technology and then talk about some new applications, new application-related products that we developed, and then end with two new DNA sequencers. One, a highly accurate short-read sequencer that produces reads that are Q40 or more accurate, and then a new high-throughput long-read sequencer we call the Revio System. So first, the PacBio technology. So PacBio has commercialized a technology called HiFi Sequencing that produces highly accurate long reads. The fundamentals are observing a DNA polymerase synthesizing a single individual native DNA molecule in real time, incorporating fluorescently labeled nucleotides. The label of those nucleotides denotes the identity, the ASHG or T, and then the kinetics, how fast the polymerase moves, is associated with epigenetic marks like DNA methylation. So you can directly observe for very long DNA molecules the sequence accurately and the methylation status of all the CPG sites. This occurs on a device we call a smart cell, so you look at millions of molecules in parallel. This technology has been used to produce extremely complete and accurate genomes, transcriptomes, and epigenomes. It was an important part of the first telomere-to-telomere assembly of a human genome. It was used to produce the first catalogs of not just genes, but full isoforms from 5' to 3' at the single cell level, and then to produce directly phased methylomes. So you observe in the same molecules, methylation status, and haplotype. PacBio also has a highly accurate short-read sequencing technology called sequencing by binding, which achieves that order of magnitude around 15 times fewer errors than you get with sequencing by synthesis. And this is based on a chemistry that separates the interrogation of the next base from the incorporation and the extension. So you interrogate with fluorescently labeled bases, but then those are removed, and you incorporate a native nucleotide. So with these technologies, our focus for the last year has been in three areas. The first is to expand and improve the applications around HiFi sequencing. The second was to develop a sequencing by binding platform to actually release that to the market. And the third was to develop a new platform that would address the two most common requests for HiFi sequencing, which is improved cost and increased throughput. So first on the applications. Over this year, we've released some new library prep kits and automation of those kits to make it easier to prepare libraries for PacBio sequencing. These, in total, have reduced the amount of DNA required for a library by 40%. It's reduced the hands-on time by 60% for manual preps, and much more if you're using the automated prep, of course. And then reduced the number of tubes required by 30%. We've also released DNA extraction kits using a technology called nanobind that extracts extremely high molecular weight DNA. That's excellent for our long-read sequencing products. And then we extended our platform to provide that methylation capability I was talking about comes out in every run. So without any special library prep, you get the methylation calls along with your standard DNA sequencing runs. Just want to highlight at this conference, there's some continued improvements, some of the early work to continue to improve the HiFi workflows being presented here. One, there's a poster from Chris Saunders talking about improved models for 5-methylcytosine calling and the ability to go from calling what we currently do, which is CPG sites, to call hemi-methylated sites or methylation in non-CPG contexts. There's also work from Julian Rocha to take the automation that I talked about, where you start with sheared DNA and prepare libraries, and to extend that to do automation of the extraction and the DNA shearing all on the same robot. And then finally, from Juniper Lake on improved variant calling methods to get more accurate variant calls out of the same HiFi data. Now on the application side, there's two products that we released this week. We released recently. The first was panels for targeted sequencing in collaboration with Twist. So in particular, there are two panels. One, a pharmacogenomics panel that targets 24 genes of interest, and I'm sorry, 50 genes of interest. And then there's a second one, which is a dark genes panel targeting 389 genes that are difficult to sequence with short read sequencing and where the long reads add value. So these panels basically let you do what you can normally do with targeted sequencing, which is focus on the areas of interest and get less expensive and higher throughput sequencing from the same platform. So on the Sequel IIe system, with a pharmacogenomics panel, you can do 24 samples on a single SMRT Cell. And for the dark genes panel, you can do 4 samples per SMRT Cell. So as I mentioned, this is a product released in partnership with Twist. And this is taking advantage of the complementary strengths of the two companies and the two technologies. So Twist provides double-stranded probes that have very high capture efficiency and provide exceptional uniformity of capture across different CG contexts. And PacBio has the complementary technology to sequence those fragments and to provide extremely uniform sequencing. The panels, the long read panels, are a modified and optimized version of the capture probes that were used for short reads. So these panels have been optimized to use fewer oligos. You don't have to be as dense because you're capturing bigger pieces, and to optimize conditions to give as uniform GC balance as we can achieve, and to capture fragments that are longer than 5 kb. So Twist has the technology and supports the panel design and the library prep and target enrichment, and then PacBio supports the SMRTbell library prep and the sequencing side. To show how this data look, this is showing the pharmacogenomics panel with 24 samples here multiplexed on a SMRT Cell. So you see on the left the read length distribution, and the reads are colored here by whether they're on target or off target or duplicates. So about half the reads end up on target, which is a 784-fold enrichment over if you had done whole genome sequencing. So the capture is working very well, leading you to get coverage on average of these 24 samples of 190-fold over these genes of interest. And then you can see on the right here how the reads look. This is a 42,000 base pair region. So you can see very uniform coverage across these 24 samples, across the full 42 kb. In terms of what this costs per sample, so on the Sequel IIe system, doing 4 samples per SMRT Cell, the library prep and sequencing reagents cost $315 a sample, and for the pharmacogenomics panel, it's where you multiplex higher, it's $50 a sample. The second application kit that we released is around RNA sequencing, specifically what we call isoform sequencing or IsoSeq. So what's unique about PacBio RNA sequencing is you don't just read individual fragments. You see the entire RNA isoform, the spliced isoform, from 5' to 3', the entire molecule. And it's that combination of read length and high accuracy that makes PacBio HiFi sequencing a great fit for single-cell RNA sequencing. You can see the full isoform, and you have the accuracy to detect barcodes. The real limitation of HiFi for RNA sequencing has always been around the number of reads you can get. So in a typical PacBio sequencing run, you get a few million reads, single-digit million reads. But there's a technology developed out of the Broad Institute last year and now commercialized in partnership with PacBio, which is the MAS-Seq method. And this method increases throughput through concatenation. And basically the idea here is rather than just sequencing an individual 500 or 1,000 base pair isoform, you concatenate a bunch of them together into a longer molecule with known spacers, so you can then pull it back apart after sequencing. So now, rather than getting only one read or one RNA per read, you can get 15 or more. The product for this is called the MAS-Seq for 10x Chromium single cell 3' kit. So as its name suggests, you do your single cell, you make a single cell library using the 10x Chromium single cell 3' kit, and then use this MAS-Seq kit to do the concatenation, followed by sequencing on the PacBio platform. This kit has a number of improvements over the academically published protocol that I mentioned, the so-called homebrew solution here. Well, one, it provides all the reagents, all the oligos, and other reagents in a single kit. It's also improved the workflow to cut about a day off of the prep time. There's a software that we provide to do the deconcatenation, and then we're providing about a third more reads per run. So you get now for a single SMRT Cell, you can get around 30 to 40 million reads. To show how that data look, this is 2 runs for peripheral blood cells where there was a single cell prep done on the with the 10x instrument. Then a MAS-Seq library was prepared, and after deconcatenation, you end up with 40 million reads in one run and 43 million in another. And then you can do the kind of analysis you'd be accustomed to, where single cell, where you'd look for clusters of different cell types, and you can see those clear clusters. But now, rather than these clusters being built from small snippets of individual genes, they are from individual isoforms. You can see cell-specific isoforms. So this kit is available to order today. It will begin shipping in early November. The price for this, a single kit has 8 reactions, and the cost is $475 a sample. And you can see up here the kind of breakdown between the 10x genomics cost and then the library prep cost and sequencing. So in total, for a PacBio MAS-Seq experiment on the Sequel IIe, it's around $3,000. And this is the only solution to get this one technology solution for the full isoforms with supported software and with high accuracy. Okay, so moving on from the applications then, the other 2 key products that we've announced this week are one, a new DNA sequencer that commercializes the sequencing by binding technology. The way we've been thinking about that is we've had HiFi sequencing, which delivers long and highly accurate reads, even in hard-to-sequence regions of the genome, and this can be useful for many applications. For some applications, it's a great fit. For others, it's less ideal. And now with sequencing by binding, we have a complementary technology that can be a fit where HiFi is not. So rather than trying to force the problem into the sequencing technology, we can offer the right tool for the problem. If you need long-range information, if you need methylation, HiFi sequencing is the technology for that. If you need a large number of counts or extreme accuracy to look for mosaicism or things that are at low frequency, it's sequencing by binding. We first started talking about sequencing by binding at AGBT this year and talked about what I showed earlier, the fundamentals of how it works, a demonstration that you can get 15 times higher accuracy per base than you can get with sequencing by synthesis, and then some examples of that and other benefits of the platform. But now we're able to talk about the instrument that actually will make this available to you. It's called the Onso. The key specs of this platform is that it offers 400 to 500 million reads per run, and it will provide a 200 cycle kit, so offering 1 by 200 and 2 by 100 base pair reads. There will also be a 300 cycle kit that offers 2 by 150 reads. As I mentioned, the real hallmark of this sequencer and what makes it unique is that 90% of the bases are Q40 or greater. So this is an error rate of 1 in 10,000 bases or lower. We also offer conversion kits from existing short read libraries, so you can take a P5, P7 library, convert it to an Onso library. To show some examples of what you can do with this high level of accuracy, we've applied the sequencing by binding to the SaraCare control samples, where you can spike in variants at known low frequencies and look at your sensitivity for detecting those variants. So what you see here is on the x-axis, different variant frequencies where it's spiked in at anywhere from 0.5% to 0.05. And then the pink line is sequencing by binding, and the orange line is sequencing by synthesis. So you can see at similar read counts. So in this case, it's 6,000 reads of each, or 6,000 X coverage, I'm sorry, of each. That at all variant frequencies, the sequencing by binding has higher sensitivity. And just to show why that is and where that comes from. So if you imagine looking, or this is the data, looking at sequencing by synthesis at one of these positions with the spiked in variant, that variant's been spiked in at 0.1% frequency. And it's specifically the wild type allele is a T, and the spiked in variant is a C. So if you look at the sequencing by synthesis reads, you'd see that, yeah, sure enough, most of them showed the T, 99.7% of them showed the T. But then you see at low frequencies, both the A, C, and G. And they're at a level where it's not possible with high confidence to say that this C is in fact biologically there while the A and G aren't. If you look instead at similar coverage for sequencing by binding, you see now that, again, most of the reads come from the T, And now you see there are 6 reads that have the C, so right around that 0.1% frequency, and you don't see the A and G ever. So this is showing what that extreme level of accuracy lets you do. You can really tell that, yes, there is a low frequency variant here, this C variant. Now, the way this is often addressed with sequencing by synthesis is to add unique molecular identifiers and to sequence to even higher depth, and then correct the reads that have the same unique molecular identifier. So this is now taking that similar data set and using the unique molecular identifiers to correct the sequencing by synthesis reads. And now you're sequencing to 4 times higher depth here. And comparing to sequencing by binding without unique molecular identifiers, so without the complexity of the workflow to add those, and without the additional sequencing costs. And you can see even at this level, the sequencing by binding is producing cleaner results than the corrected sequencing by synthesis. So this platform is now in beta testing. It's in beta testing at 3 locations: the Broad Institute, Corteva Agriscience, and Weill Cornell Medicine. And we will be accepting orders for the system and in the first quarter of next year, and plan to ship in the first half of next year. All right, I'll see if I can get in before the hall closes. Our last product, which is a new long-read sequencing platform to address the 2 most common requests we get around HiFi sequencing, which is increasing the throughput and lowering the cost. So as I said, HiFi sequencing is known for producing extremely accurate and complete genomes, transcriptomes, and epigenomes. With the real challenge is always being around scale. And we're addressing that with the system we call the Revio System. And this system offers HiFi sequencing at scale. So in a single run, you're able to interrogate 100 million ZMWs, which compares to 8 million on our current system. And the sequencing runs are 24 hours, producing 360 gigabases of HiFi yield per run. This is while maintaining the read length and accuracy that we offer on the Sequel system. So you get the typical average read lengths will be 15 to 18,000 bases. The 4 key benefits of this system are at the scale, as I mentioned. So it's offering this 100 million ZMWs per run through 4 independently operated 25 million ZMW smart cells. So you can run anywhere from 1 to 4 smart cells per 24 hours, offering up to 100 million ZMWs. These 3 improvements, the denser smart cell, the ability to run 4 at a time, and the 24-hour movie times, which is down from 30 on the prior system, in total is a 15x increase in throughput from the Sequel IIe. The system's also been designed to improve ease of use to support operating the system at that scale. We've reduced the number of consumables by 50%, so now you just load a single tray and a single plate on the system to initiate a run. We offer the ability of what we call loading runs in advance. Basically, it lets you set up the consumables for your next run while the current one is going on. So you can make sure that the system always has the next run to go without you having to be there right when the instrument completes. We've also modified the smart cell design and the chemistry. So there's no longer a need for an external nitrogen supply, which is a requirement of the Sequel IIe system, and that makes the lab setup easier. A third key benefit of the Revio system is increased compute power. In total, we offer 20 times more compute power on this system through the addition of GPUs. This has allowed us to do a lot more things on the instrument. One of the more notable ones is bringing on an algorithm called Google Deep Consensus. And this is a deep learning algorithm that improves the yield and accuracy of the data you get off a HiFi sequencer. So now this is included as part of every run. We've also improved the affordability of the system. So we're offering a $1,000 human HiFi genome. So it's current the consumable pricing for doing a single smart cell 8M is $995, and the yield of that smart cell is 90 gigabases, a 30x human genome. We've also improved affordability through these ease of use improvements. It's now possible to load a run in under a minute. So you've reduced the labor time, and then we've decreased the file size off the instrument by 50% per base, so it costs less to store the data as well. As I mentioned, there's a 15x improvement in yield for a total of 1,300 HiFi genomes a year. 1,330x HiFi genomes a year. Of course, if you're operating at a different coverage level, 10x coverage, you'd be at 3,900 genomes a year. Let's talk a little bit more about how the system achieves this capacity. One of the key components of the system, as I mentioned, is the smart cell. The 25M smart cell is physically effectively the same size as the smart cell 8M, and it takes a lot of lessons for how we manufactured that consumable part and brought it into this 25M. So we were able to take advantage of the ability to manufacture these efficiently, but at the same time improving the key functions of this, or the key attributes of the smart cell, specifically the increased density. So we have 3 times more spots per unit area. We've also improved the loading and illumination uniformity. It's a bit hard to see in this picture, but there's a cap over the system, a plastic cap that controls the flow of reagents over the cell. That has a few key things. One, it improves the loading uniformity. It also reduces the volume of reagents that need to be pipetted onto the chip. And then this is part of the ability to do nitrogen-free sequencing. The flow cell cap prevents the open flow of air over the smart cell and removes the need for nitrogen. As I mentioned, we have simplified the consumables and the user experience. So what used to be 5 parts on the old system is now 1. And all you have to do to set up a run is put on that the smart cell tray and the single reagent plate. The reagent plate now contains this sample. So you'll put your sample in there, put that single plate in one of the reagent deck locations, put the smart cells over there, and start the run. The ability to run 4 smart cells at once comes from a change in the architecture of the system, where now there are 4 independently operable stages. So this is kind of a zoom-in view of what's behind the door in the instrument. There are these 4 different stages where you can you independently look at a smart cell. That means you can run different libraries on the different smart cells. You could run whole genome sequencing on one, RNA sequencing on another, or any combination thereof. The compute power, as I mentioned, it's 20x times higher compute power on this new system, largely through the inclusion of GPUs. We have NVIDIA GPUs on the instrument, which has enabled the Google Health Deep Consensus to be brought on board. And then we've also optimized the BAM file format to reduce the file sizes by around 50%. So we've been running these systems at PacBio for a number of months, and it's just showing some representative recent runs. Of course, the most interest here at ASHG is human samples. Those top samples are a bunch of human runs, and then some animal and plant samples showing consistent performance across all these samples with the 90 gigabases or more per smart cell, and then more than 90% of bases Q30 or above. I think it'd be interesting to see the data, and this is just showing a human sample, one of the genome in a bottle samples, HG002, sequenced on the Revio system on the top and the Sequel IIe system on the bottom. So you can see we have the long, accurate HiFi reads that allow the haplotypes to be phased. So you can see the bottom haplotype, which looks like the reference, and the top haplotype that has a number of variants. These look identical between the Revio and the Sequel IIe. You can see the high accuracy and consistency of the 2 platforms. And then on the right-hand side are showing these same reads, now with the methylation information layered on. And this, let me close this here, apologies. This is a parentally imprinted gene. So here the paternal allele, which is the bottom one, is expressed, and the maternal allele is silenced by methylation. So you can see here the red showing the methylation and the blue showing the unmethylated. So this combination of long, accurate reads, phasing, and methylation, and consistency between the 2 platforms. It's not just the raw data that looks comparable between the 2 platforms. The Revio also has excellent application performance. It's matching the high variant calling performance of the Sequel IIe across single nucleotide variants, indels, and structural variants, and then providing excellent genome assemblies. So this is showing genome assemblies for 3 of the human samples I showed a few slides back, a non-human mammalian sample, an insect, and a plant sample. You see, for all these, you're getting extremely long assembled pieces. To tie back to one of the applications, the Revio is also providing a boost not just for whole genome sequencing, but for all the applications. For this dark genes panel I mentioned, you could do a 4-plex on the Sequel IIe system. Now on Revio with the 3 times more ZMWs per smart cell, you can do 3 times as many. You get 3 times as many reads. You can do 3 times as many samples. And this is now showing a 12-plex of the dark genes panel. And you can see you're getting comparable read lengths on the Revio system, comparable read accuracies, and the same on target rates and folder richness. And now it's now showing the uniformity of coverage for 12 samples for this dark genes panel. That, of course, has an impact on cost. So just as whole genome sequencing cost is much less, so is the targeted panel cost. So now you can fit around 3 times as many samples per smart cell, so your costs go down by about 1/3. These 2 systems, you can actually demo both of them at the PacBio booth. It's the big pink booth back there, so you can come and see them and interact and see what it's like to run these instruments. The Revio system is available to order today. The system itself costs $779,000 US list price. The consumables to do a run are $995 a smart cell, which is 30x human genome. So it's under $1,000 consumable costs to do a genome on the Revio. And as I mentioned, you can do 1,300 genomes a year, 1 to 4 genomes or 1 to 4 smart cells or genomes in parallel, 24-hour runtime. The Onso system is also available to demo at the PacBio booth. It will be available to order in the first quarter of next year. And this system, again, offers 400 to 500 million reads with either 200 or 300 cycle kits, 48-hour runtime, and greater than 90% of bases Q40 and above. So with that, I think we have 2 minutes until the hall closes, but happy to take any questions. Thank you for coming.